Hello everyone, this is Dr. Meghna, your dentist. Welcome to the Dent Guide. I'm going to discuss with you the most important topic in periodontics, the dental plaque. So without wasting a moment, let's get into the video. Let's see what biofilm means. Biofilms are composed of microbial cells encased within a matrix of extracellular polymeric substances such as polysaccharides, proteins, and nucleic acids. Let's see the functions. What benefits does bacteria have living in a biofilm? Let's see one by one. This is important. It can come as a very short answer question in your university exams. First one, resistance to antimicrobial agents. Biofilm bacteria are often up to 1,000 times more potent to antimicrobial agents than their planktonic counterparts. What are planktonic species? These are free floating in saliva and gingival crevicular fluid. Bacteria that grow in a multi species biofilm interact closely with neighboring cells. Sometimes these interactions are mutually beneficial, as in the case when one organism removes another organism's waste products and uses them as an energy source. In other instances, bacteria compete with their neighbors by secreting antimicrobial molecules such as inhibitory peptides and hydrogen peroxide that might kill the other species. The biofilm mode of growth facilitates cell-to-cell signaling and DNA exchange between bacteria. Biofilms frequently contain microcolonies of bacterial cells, and these can form a primitive circulatory system. This primitive circulatory system removes waste products and brings fresh nutrients to deeper layers of the biofilm. The biofilm matrix also functions as a barrier. Substances produced by bacteria within the biofilm are retained and concentrated, which fosters metabolic interactions among different bacteria. Let's see the criteria of biofilm. How do bacteria function in the biofilm? The first is autopoiesis. Autopoiesis is the ability to self-organize. The bacteria within the biofilm can adapt to environmental conditions and they can self-organize. The next is homeostasis. Homeostasis is the ability of the bacteria in the biofilm to resist environmental perturbations. What is synergy? Synergy means interactions. The bacteria interact together and function as a single unit. What is community? This is the ability to respond to environmental changes as a unit. These are the functions of biofilm. This is the criteria for a biofilm. The biofilm question can come as a very short answer question or a short answer question in your university exams. So pay attention to it, read it up. Let's see what materia alba is. You can see it in the diagram. It is loosely attached biofilm that can be displaced easily. It consists of soft accumulations of bacteria, food matter, and tissue cells. Let's see the exact definition from Carranza. Materia alba refers to soft accumulations of food, bacteria, and tissue cells that lack the organized structure of dental plaque and that are easily displaced with a water spray. Here are the important differences between different types of biofilm: the materia alba, the dental plaque, and the calicculus. Let's see each one in detail. What is materia alba? It is a white cheese-like accumulation. Let's see plaque. It is resilient, clear to yellow-grayish substance. Calicculus is hard deposit that forms via the mineralization of dental plaque. Materia alba is a soft accumulation of salivary proteins, some bacteria, epithelial cells, and occasional disintegrating food debris. It lacks the organized structure and is therefore not as complex as plaque. It can be easily displaced with a water spray. Dental plaque is composed primarily of bacteria in a matrix of salivary glycoproteins and extracellular polysaccharides. I'll discuss dental plaque and calculus in detail in the coming session. It is considered to be a biofilm. It is impossible to remove by rinsing or with the use of sprays, while material alba alone can be removed. Calculus generally is covered by a layer of unmineralized plaque. Hope these differences are clear. This is important. Note it down. Let's see what dental plaque is. The definition, the composition, the formation of plaque, and the theory supporting the formation of dental plaque. Let's get into the definition. Dental plaque is defined clinically as structured, resilient, yellow-grayish substance that adheres tenaciously to intraoral hard surfaces, including removable and fixed restorations. This is taken from Carranza. Extremely important. The dental plaque can come as a long answer question or short answer question. Let's see the composition of dental plaque. 1 gram of plaque contains 2 into 10 power 11 bacteria. See how huge the bacteria number is. The supragingival plaque alone contains 10 power 9 bacteria. The periodontal pocket, if it's a healthy crevice, it contains 10 power 3. If it's a deep pocket, it contains more than that in the healthy crevice. Plaque is a mixture of microorganisms and matrix. What microorganisms? They can be bacterial, rods, cocci, spirochetes. What are non-bacterial here? Archaebacteria, yeast, protozoa, and viruses can also be found in dental plaque. Let's see the matrix of dental plaque. It contains primarily of water. Water is the major component. The others being organic and some are inorganic. Let's see the organic matrix. It consists of polysaccharides, proteins, lipids, and glycoproteins. Polysaccharides account for almost 30% of organic matter. Polysaccharides are produced by bacteria. What are these polysaccharides? Usually dextran, levain, galactose. Proteins account for 30% of the organic matter of matrix. These proteins may be albumins. The source of albumins is from gingival crevicular fluid. Lipids account for 15% of the organic matrix. Lipids come from the membranes of disruptive bacteria or host cells. Membranes are nothing but phospholipids, right? Glycoproteins form the pellicle. These glycoproteins are from the saliva. Let's see the inorganic matrix. Usually there are two types of plaque, supragingival and subgingival. The constituents of supragingival plaque come from saliva. The constituents of subgingival plaque come from gingival crevicular fluid or GCF. The organic components are calcium, phosphorus, and traces of sodium, potassium, and fluorine. I have made this pictorial representation of plaque biofilm. These are the rods. This is the extracellular matrix. These are the primitive water channels. And the matrix contains cocci, rods, and spirochetes. You can make this simple diagrammatic representation of plaque biofilm in your exams. Let's get into the site specificity of bacteria in plaque. Supragingival plaque typically demonstrates the stratified organization of a multi-layered accumulation of bacterial morphotypes. The gram-positive cocci and short rods predominate at the tooth surface, whereas gram-negative rods and filaments as well as spirochetes predominate at the outer surface of the matured plaque mass. In general, the subgingival microbiota differs in composition from supragingival plaque primarily because of the availability of blood products and a low reduction oxidation or redox potential which characterizes the anaerobic environment. Also, the availability of GCF nutrients to bacteria in subgingival plaque. Subgingival plaque is of two types. It is tooth associated and tissue associated. At this location, the filamentous microorganisms dominate, but cocci and rods also occur. Plaque is dominated by gram-positive rods and cocci, including Streptococcus mitis sanguineus, actinomyces species like Actinomyces aureus. Eubacterium species may also be found. However, in deeper parts of the pocket, the filamentous organisms become fewer in number. In the apical portion, they seem to be virtually absent, no filaments. But gram-negative rods might be present. The tissue-associated subgingival plaque is associated with crevicular epithelial cells. In this, both host tissue cells and bacteria like Streptococcus species, Peptostreptococcus, P. gingivalis, P. intermedia, Turnarella, and Fusobacterium species might be found. Let's get into the classification of dental plaque. This can come as a short answer question. First classification. On the basis of position on the tooth surface toward the gingival margin, it is classified as supragingival and subgingival. Let's see what supragingival plaque is. Plaque at or above the gingival margin when in direct contact with the gingival margin. What is subgingival plaque? Plaque found below the gingival margin between the tooth and the gingival pocket epithelium. The second classification is the Grant's classification. The first classification is taken from Carranza. The Grant's classification is based on location. Coronal plaque. The plaque that is coronal to gingival margin. Gingival plaque. It is on the external surface of oral epithelium and attached gingiva. What is subgingival plaque? It is below gingival margin and near to junctional epithelium. What is fissure plaque? The plaque on the pits and fissures of tooth. What is peri-implant plaque? Plaque on the implant surface. This is responsible for causing peri-implantitis, an inflammatory condition associated with implant. You can see this flowchart. You can write this flowchart when the classification is given. Plaque is two types: supragingival, subgingival. Supragingival is again divided into coronal and marginal. Coronal is above or coronal to gingival margin. Marginal plaque is at the level of gingival margin. Subgingival can be divided into unattached and attached. Attached is subdivided into tooth associated and tissue associated. The unattached plaque, you can see this. This is the unattached plaque. This is responsible for causing gingivitis. The bacteria in this plaque will affect the gingiva here. Next is tooth associated plaque. This is the tooth surface and this is the tooth associated plaque. This is responsible for calculus formation and dental caries. Since it is closely associated with the tooth, it causes calculus formation and caries. Next is the tissue associated plaque. This is the tissue and this is the tissue associated plaque. This is responsible for causing gingivitis and periodontitis. It can go in contact with the bone too. You can see bacteria in the connective tissue, bacteria in the bone, bacteria associated with the epithelium. Hope this classification is clear. Let's get into the formation of dental plaque biofilm. There are three main phases. First, formation of pellicle on the tooth surface. Second, initial addition or attachment of bacteria. Third, secondary colonization or plaque maturation. Let's see each one in detail. What is acquired pellicle? All surfaces in the oral cavity, including hard and soft tissues, are coated with a layer of organic material known as acquired pellicle. The pellicle on tooth surfaces consists of more than 180 peptides, proteins, and glycoproteins. These glycoproteins may be keratins, mucin, phosphoprotein, proline-rich protein, histidine-rich proteins. These function as adhesion sites or receptors for bacteria. The salivary pellicle can be detected on clean enamel surface within 1 minute after the introduction into the mouth of volunteers. By 2 hours, the pellicle is essentially in equilibrium between absorption and detachment, although further pellicle maturation can be observed in several hours. The salivary proteins form a lubricating layer and this is called as acquired pellicle. This pellicle can be formed on mucosa and teeth. Mucosa contains epithelial cells which shed continuously. This prevents bacterial buildup. While teeth are non-shedding surfaces, they promote accumulation of bacteria. Let's see the second step. Initial adhesion and detachment of bacteria. After few hours of pellicle formation, bacteria starts to colonize by Brownian motion or chemotactic activity. Brownian motion means they come based on the concentration of bacteria available or substances available. It's a kind of diffusion. Chemotactic activity. There are certain chemicals in the oral cavity that attract certain bacteria. These bacteria, which are the first to colonize, are called initial or primary colonizers. Mostly gram-positive facultative aerobes are the initial or primary colonizers. They may be cocci or rods. Let's see the different types of primary colonizers. In rods, we have Actinomyces species, Actinomyces israeli, Odontolithecus, and Oralis. The second is AA, your favorite, Aggregatibacter actinomycetum comitan serotype A. Next is Capnocytophagus species. Let's see the cocci. Streptococcus species. What Streptococcus? Streptococcus mitis, sanguis, intermedius, and oralis. These bacteria are extremely important. You have to memorize. You have to write it down. You have to memorize. The primary organizers come and attach to the tooth surface. Initially, this attachment is physical or reversible. They firmly adhere to the pedicle in a later stage. The bacteria have adhesins or bacterial proteins. These are the adhesins. You can see this pictorial representation. They attach to proline-rich proteins or receptors in the pellicle. This is the pellicle which is attached to the tooth surface. These are the proline-rich proteins or receptors of the pellicle, and these are the adhesins, which are the bacterial proteins. The bacteria come and adhere to the receptors on the pellicle. Pellicle serves as mode of attachment to certain bacteria. The initial colonizers derive nutrition from glycoproteins and simple sugars. They eat and multiply. They proliferate. The Streptococcus species produces extracellular polysaccharides. These extracellular polysaccharides are important for adhesion and attachment. These are also responsible for initial caries. What are these extracellular polysaccharides? These are glucans and fructans. What do? They promote further adhesion of certain bacteria. They also serve as energy source. These primary colonizers, they are facultative aerobes or facultative anaerobes. They primarily feed on oxygen and deplete it. They increase the growth of obligate species. As there is no oxygen here, the obligate species are enough to colonize. The last step in plaque formation is secondary colonization and plaque maturation. As the environment becomes anaerobic, gram-negative rods colonize plaque's outer surface. These are called secondary colonizers because they come after primary organizers. The primary organizers deplete the oxygen and the environment becomes anaerobic. Now the secondary colonizers are the heroes in this. What are these secondary colonizers? They are Campylobacter species, Porphyromonas, Prevotella, Fusobacterium, Turnerella, Aggregatibacter actinomycetum comitan serotype B, Treponema denticola. These secondary colonizers adhere to primary colonizers via receptors. This is called coadhesion. This is your substrate or the pellicle contains glycoprotein matrix. The primary colonizers have depleted the oxygen and now the secondary colonizers. This is the secondary colonizers. They are attached to the substrate. This is the secondary colonizer. Now the primary colonizer comes and attaches to secondary colonizer. This is called co-addition. Let's see what co-aggregation is. When many distinct bacteria adhere to each other and form clumps, this is called co-aggregation. There is no substrate. The primary colonizers go and attach to the secondary colonizers without a substrate. Co-addition occurs in presence of substrate. Co-aggregation occurs in the absence of substrate. Let's see the two types of co-aggregation. One is corn cob formation and the other is test tube brush formation. Fusobacterium co-aggregate with all other human oral bacteria, whereas Veillonella species, Capnocytophagia and Prevotella bind with Streptococci and Actinomyces. Well-characterized interactions of secondary colonizers with early colonizers include the co-aggregation of Fusobacterium nucleatum with Streptococcus sanguis. You can see this is a rod and it is surrounded by cocci. It gives the appearance of a corn cob. This is a spirochete and it is attached to several rods. This gives the appearance of a test tube brush. Co-aggregation between different gram-negative species is likely to predominate. Mostly, this co-aggregation occurs between gram-negative species itself, gram-negative bonds to gram-negative, and less to gram-positive. So, the common examples of this co-aggregation are Fusobacterium nucleatum with P. gingivalis or Treponema denticola. In corn-cob formation, in which Streptococci adhere to filaments of Cornybacterium nucleatum. In test tube formation, this is the filamentous bacteria to which gram-negative rods usually adhere. Let's see what quorum sensing is. This is an important, very short answer. Do note it down. In a biofilm, bacteria have the ability to communicate with each other. The bacteria usually talk in the biofilm. Bacteria secrete signaling molecules which accumulate in the local environment. Those signaling molecules get concentrated in this environment in a biofilm. This triggers response after reaching a critical threshold concentration. This critical threshold concentration is reached only at high cell density, only if that particular species of bacteria are high and every bacteria is secreting the same signaling molecules. This critical threshold concentration is reached. They help the bacteria to sense that population has reached its critical mass or quorum. What is the function of this quorum sensing? They need it to increase the growth of its beneficial species and decrease the growth of its competitors. Also, they aid in the modulation of expression of genes for antibiotic resistance. An analysis is made from more than 13,000 plaque samples that is associated with subgingival microorganisms using a DNA hybridization methodology. They came up with color-coded complexes of periodontal pathogens. This is important from competitive exam point of view and can also come as a short answer question. The composition of different complexes was based on the frequency with which different clusters of microorganisms were recovered and the complexes were color-coded for easy conceptualization. The early colonizers are either independent of defined complexes like AORS, Actinomyces aureus, or they are members of yellow or purple complexes. Yellow complex consists of Streptococci, purple complex consists of Actinomyces or Enterocolyticus. The microorganisms primarily considered secondary colonizers fell into the green, orange, and red complex. These green, red, and orange complex they come under secondary colonizers, and this yellow, this independent one, and violet complex comes under primary colonizers. The green complex includes E. corrodens, C. gingivalis, Aggregatibacter actinomycetum comitant serotype A, and Capnocytophagia species. The orange complex involves Fusobacterium, Prevotella, and Campylobacter. The green and orange complexes include species recognized as pathogens in periodontal and non-periodontal infections, while the red complex consists of P. gingivalis, Tenerella forsythia, and Treponema denticola. This complex is of particular interest because it is associated with bleeding on probing. That's why the red color. The existence of complexes of species in plaque is another reflection of bacterial interdependency in the biofilm environment. As I told you, the early colonizers are either independent of defined complex or they belong to yellow or purple. The secondary colonizers are from green, orange, and red. You have to note down the important bacteria. Hope this is clear. Let's see the plaque hypothesis in another session.